Thank you everybody for coming. I hope everybody can see my slides. I am going to talk to you today about some next generation microneedle technologies that can be used for drug and vaccine delivery. And what I will focus mostly on are their applications in long-acting drug delivery, particularly of high doses of drug that we've delivered. And I'll start off by talking about the issues that I see with current systems for long-acting delivery. And then I'll move on to talk about dissolving and biodegradable microneedle systems, and then concentrate in on technology that is unique to my laboratory here at the School of Pharmacy at Queen's, hydrogel forming microneedles. And what I hope to show you for both main platforms is that we have a degree of flexibility in design, and that this creates great utility and allows us to deliver a wider range of molecules into and across the skin for high dose and long acting delivery. But as with any delivery system, microneedles need to be able to translate it, because as a pharmacist, what I want to see is patient benefit. And I'm going to talk a little bit about how we can overcome current challenges to move towards the clinic. So we think first about long-acting drug delivery. This basically involves slow absorption of a drug and the systems we can use to allow that. So if we think about why might we want a drug to be slowly absorbed into the body? So there are many chronic conditions where people have to take medication on a daily basis. Now that might be one tablet a day for hypertension or hypercholesterolemia, but it may be multiple tablets to treat an infectious disease like HIV or tuberculosis. And those people are being treated with those medications often for the remainder of their lives, as in the case of hypertension and hypercholesterolemia and HIV. But the same would be true for chronic disease like schizophrenia, for example. And if a person is continually taking a medication, that can have a psychological impact on that person. So every time they take the tablet, that can be a reminder that they are not well. And that can lead to what's known as pill fatigue. So medication has maybe reduced their symptoms or controlled an infection, so the person feels physically okay, but they're having this mental disturbance from having to take medication every day. And then they just decide to stop taking it. And that's where we can have problems. So we could have a rebound of hypertension or hypercholesterolemia leading to cardiovascular issues. Or if it's an infectious disease, we can lead to a resurgence of that disease. And we may end up with the likes of HIV coming out from viral reservoirs and actually then being resistant to future treatment with the same drug, and the regime then having to be changed. But if we could find a way to deliver the same drug on an infrequent basis, then we would potentially reduce this whole issue of pill fatigue, and we may then control conditions better and improve therapeutic outcomes for patients. The problem with long-acting drug delivery systems as they stand is that they tend to be quite invasive and require administration by a healthcare professional. So we might have something like a long-acting injection that's based on either a poorly soluble drug that slowly dissolves from particles that are deposited in the subcutaneous or intramuscular tissue. We might have a prodrug of our molecule, and we've dispersed that in oil, and the prodrug is slowly released from the oil into the interstitial fluid in a muscle, for example, and then the ester is cleaved to reveal the active drug. Or we might have particles made from a biodegradable polymer like PLGA that will control the release of the drug as it biodegrades. Or we may have an implantable system, a monolithic rod, for example, that will sustain the release of the drug over time. All of those require a healthcare professional, and all of them are invasive. And if you have an implant that doesn't dissolve or biodegrade in the skin, then that would need to be removed. So healthcare workers need to intervene, so that adds to cost. We also have as sharp to dispose of, and there is potential then for needle stick injuries. And then we have up to 20 to 30% of people who are needle phobic. So they may not even consent to these treatments that may benefit them. So we need to think about other ways, less invasive ways to deliver long-acting therapeutics. And that's where microneedle systems come into play. So as you can see from the figures here, microneedles are arrays of tiny structures. They're no more than 1 millimeter in height, so whenever we press them into the skin, they are painless and they don't cause any bleeding. But as you'll see in the rotating optical coherence tomography image here on the left, they create regularly spaced aqueous pores in the skin. And that means now we can deliver pretty much anything we want to into or across the skin. And that creates a lot of possibilities for long-acting delivery if we can use microneedles to deposit our long-acting platforms in the viable skin layers instead of using an injection or an implant. Now, microneedles traditionally have had small patch sizes, and that meant that we usually could only deliver high and pretty low doses, and that meant that microneedles were really concentrated on vaccine delivery. But what I hope to show you today is that if we can get our formulation right, and if we can consider larger patch sizes, then we have the potential for long-acting drug delivery. So the first example that I'm going to give you is dissolving and biodegradable microneedles for long-acting drug delivery. So what we would do here is we would take a nano-formulated drug, so either a poorly soluble drug nanocrystal or a biodegradable drug-loaded nanoparticle, and put those at high concentration into an aqueous gel. And we then cast that into a silicone mold and dry it to form the needles. And we can then come along with a base plate, and that will make our microneedle array patch. When the microneedles would be inserted into the skin, the water-soluble polymer that the needles were comprised of would disperse and then deposit our long-acting formulation in the skin in a pain-free, blood-free manner. The alternative is to make the tips of the microneedles from a biodegradable polymer drug solution cast from organic solvent, and then make the upper part of the needle from a water-soluble polymer. So essentially we would be depositing tiny drug-loaded implants in the viable skin layers. So what we want here is the needles to be deposited with their contents in the skin, and then be able to quickly remove the base plate upon which they're formed, and then the skin to close over and leave the slowly dissolving or slowly releasing drug in the skin for absorption by the rich dermal microcirculation. And remember, about 1/3 of our blood is flowing through our skin at any time. So once you've deposited drug in there, you've got a very rich site for drug absorption. So we first started working on long-acting delivery using dissolving microneedles in collaboration with the PATH organization. And what we did was to look at the HIV drug rilpivirine that is currently given by Janssen in a long-acting injection into the muscle every 1 or 2 months. So here we took the long-acting injection and we formulated it into a dissolving microneedle and applied it to the skin of rats. And then we followed plasma levels over a 2-month period, and we found that we still were measuring therapeutic human plasma levels in the plasma of the rat after 56 days. This was after a single 24-hour application of microneedles. We then looked at another HIV drug, etravirine, and then we found that it didn't matter whether we made a nano suspension of the etravirine or whether we just used the powder out of the bottles to mix into our aqueous gel to form the needles. We still saw therapeutic plasma levels in the rat after about a month. The microneedle patch again was only applied for 24 hours, the needles dissolved, and then the patch was removed. You can see what these microneedles look like here. They've got this very high solid content of drug at the tips, and then we have drug-free upper parts that will dissolve and leave these drug-containing parts in the skin. We got similar results with bictegravir, another HIV drug, and actually here the microneedles, whether they were a nano suspension or just the microparticles from the drug powder, they had comparable plasma levels to an intramuscular injection after 3 weeks in the skin of rats. So we then looked at this in quite a detailed way with cabotegravir. So in the ViiV Healthcare long-acting injection, we have the free acid form of the drug. And here we took that long-acting injection and we injected it intramuscularly or intradermally. We saw maintaining high levels in the plasma over a 28-day period in the rat was possible with both injections. With the microneedles, with the long-acting form, in other words, the injection turned directly into a dissolving microneedle, or the free acid form, that was the powder form of the drug, so not nanocrystals, or the more water-soluble sodium salt, we saw that the plasma levels were less than they were with the proprietary product injected intramuscularly or intradermally. But the microneedles, after a 24-hour wear time in the rats, all had still therapeutic plasma levels after 28 days. And after 7 days, we found that the microneedles were as competitive in terms of plasma levels as either injection using the more soluble water sodium salt. We've delivered numerous other long-acting, poorly soluble drugs, risperidone and olanzapine for schizophrenia, atorvastatin for hyperlipidemia, retigatin for Parkinson's, contraceptive hormones, a range of other antivirals, including for hepatitis and HIV drugs, using these long-acting microneedles where the drug poor solubility is controlling the release of the drug. And what we found was that these drugs all exhibit flip-flop pharmacokinetics, where the overall pharmacokinetic exposure is not governed by the elimination phase, but instead by the slow absorption phase. And what that means is that it makes allometric scaling to ascertain human patch size from rat data not so straightforward. So instead what we did was we worked out the amount of drug we were depositing in some excised neonatal porcine skin, which is the closest we can get to human skin in terms of barrier function and overall properties. So once we worked out how much we were delivering, we worked out how much was being delivered in the proprietary products and used that to conduct some physiologically based pharmacokinetic modeling. And what we found was that if we tried to do monthly delivery with microneedles for any of these drugs, the patch size would be enormous, probably the size of a person's back. So that's not going to be something that can be commercially translated. But we found that for a week's delivery, the patch size would be no bigger than about 30 square centimeters. So we've made patches that size, and we think that a person would probably remember to apply their microneedle patch once a week on a Monday morning, say, rather than once a month. So we wanted to try this out and see what would happen if you repeatedly applied microneedles. Could you maintain therapeutic plasma levels in the rat? So we looked at this with rilpivirine and with cabotegravir, and we had 2 different cohorts in both cases. So in cohort number 1, we gave a bolus dose of the intramuscular long-acting injection, and then every 2 weeks we applied the microneedles. And we did the same in cohort 1 with cabotegravir. In cohort 2, we just applied microneedles every 2 weeks and we measured the plasma levels. And with rilpivirine, what we found was after 2 weeks, it didn't matter whether we gave the initial dose with microneedles or whether the initial dose was an intramuscular injection. We were maintaining therapeutic plasma levels for the whole time, and it didn't matter what the initial dose was given with once we have rilpivirine as the drug. With cabotegravir, it took 28 days for the 2 profiles to align. But again, whether we injected with intramuscular injection first and then topped up with the microneedles every 2 weeks, or whether we just applied the microneedles every 2 weeks, we were able to maintain therapeutic plasma levels. And in each case, the microneedle patch was only applied for 24 hours and then was removed. So we have 2 options here. We could apply the microneedles regularly, say once a week, or we could give a bolus dose every time the person goes for a checkup, maybe every 6 months, and then we would give them a box of patches home, and then they would apply the patches on a weekly basis to top up the plasma levels. And this may be beneficial for patients in that they wouldn't be getting dosed with their drug every day. It would only be on a weekly basis, and this may improve compliance. So we then wanted to look at an antiviral or antimalarial application. and we decided we would look at chloroquine, and we would also look at primaquine in terms of delivery with microneedles. So we did some work with excised skin, and we found for both drugs with dissolving microneedles, we were able to deposit the drug in the skin, and the drug was also permeating right across the skin into the receiver compartment of the Fran cell. So we then looked in mice at the plasma levels, and we saw for chloroquine, the microneedles were fairly close to the therapeutic plasma level for 7 days after a 24-hour microneedle application. With primaquine, after 1 day, both microneedles and oral delivery had both fallen below the human therapeutic level in the mice. But we challenged the animals, and we saw with the chloroquine microneedle patch on its own, or with one chloroquine microneedle patch and a primaquine microneedle patch, that we were able to keep the parasitemia in infected animals at bay. Whereas if we looked at the primaquine microneedles on their own, or in the in control that was infected and not treated, the amount of parasitemia in the blood continued to increase. So we may be able to devise a microneedle system that could be a candidate for a once weekly treatment of malaria, but further work is clearly going to be required by us and by others here. So then we wanted to consider another tropical disease. So here we're considering leishmaniasis. So a person here gets bitten by a sandfly, and this can cause very disfiguring lesions of the face and other parts of the body, and the parasite can then contaminate the lymphatic system and cause damage to the lymph nodes. Currently, treatment is by injection of amphotericin B, and you can imagine getting intradermal injections of this liposomal formulation would be very painful using a needle and syringe. And also you would have the potential for systemic absorption of amphotericin B, which can cause problems with drug-drug interactions and organ toxicity. So here we wanted to load this drug into dissolving microneedles for local application in the skin, but we also wanted to control the particle size of the solid amphotericin B particles with a view to targeting the lymphatic system. And so it's known that if you keep your particle size below 100 nanometers, you can target the pores in the lymphatic vessels, and the skin is full of lymphatic vessels and has a higher hydrodynamic pressure than either the muscle or the subcutaneous tissue. So you could be able to target infected lymph nodes in this way. So here we see with the comparator, if we inject the ambisome intravenously, then it goes everywhere in the body, and you can see that measured in the plasma. With the local nanocrystal application of microneedles, you don't see that. In the lymph nodes, the microneedles give higher levels than the IV injection, whether that's the axillary or the lumbar lymph nodes. But it's really important that in the liver, the spleen, and the kidney, we don't see much in the way of levels of the drug. And this is particularly important in the kidney, where we know that injection of amphotericin B can cause nephrotoxicity. Here we're able to avoid that. So we get the drug where we want to in the skin and in the lymph nodes, but we avoid targeting organs where we may have toxicity, with the kidney being the one that's especially important. So now we look at another compound, flufenazine. So this is an antipsychotic, normally given as a long-acting decanoate ester in an oily injection every month. So here we wanted to have a look, could dissolving microneedles give sustained delivery? So we took the ester alone and put that into dissolving microneedles, or we made a nano emulsion from an oil and dispersed that in the dissolving microneedle tips, or we made the tips of the microneedles from PLGA for biodegradation. And what we found was that we were measuring therapeutic plasma levels in the rat for a full 14 days after microneedle application. And the microneedles were only inserted for a 24-hour period. We avoided the high peak plasma levels that you would see with conventional dosing, like oral delivery, for example, or intramuscular injections. But we were able to sustain the levels. And this might be good for schizophrenic patients who are needle phobic and refuse to have long-acting treatment as a result of that. And as a result, their condition is difficult to manage. So we may have a way of sustaining delivery of antipsychotics as well. And then we decided we'd look at another HIV drug, tenofovir alafenamide, but we would look at a drug that was water soluble and see could we control the administration of that. So if we just took the microneedles and put them into a dialysis membrane in vitro and looked at release, we could only really see release over a 48-hour period. And the same was true when we moved to the rat model. So we used a biodegradable microneedle tip, but the drug was released and cleared from the animal very, very quickly within a few days. So we weren't able to sustain delivery. And the reason for this was to maintain a reasonable patch size, we had to put quite a lot of drug into the PLGA tip of the microneedles. And that meant that instead of biodegradation of those tips controlling drug release, the amount of drug that was in the little micro implants was controlling release. So tenofovir alafenamide was dissolving from the surface of the PLGA tips into the interstitial fluid, and that was allowing fluid to get into these PLGA tips, dissolve the drug out, and then we had just a series of little ghost ships floating around with no drug in them. And so what I would say is for a fairly high dose water soluble drug, microneedles probably aren't going to be most suitable if they're dissolving in the skin or have biodegradable tips. You're not going to be able to control the administration for a water soluble drug over a long period of time using dissolving microneedles. What you could do though is control the delivery of a potent molecule like a hormone. So finasteride might be an example here. And so what we did was we took finasteride powder and mixed it into dissolving microneedle matrix, and we found that we would see release into the receptor compartment of a Fran cell across excised skin to a much greater extent than we would if we were to use a biodegradable needle tip made from PLGA. Because here the drug loading doesn't need to be high, the biodegradation of the implant tip can control drug release rather than the amount of drug just dissolving out, controlling release as it did with the more water soluble compound. But we would need to test this in an animal model to see what is the duration of drug release. But actually a number of other workers have shown that release over many weeks and even months of contraceptive hormones using a similar concept in animals in vivo. So then we decided let's look at a potent water soluble drug, so maybe a model protein. So here you can see that we can localize fluorescent protein in the tips of dissolving microneedles, and the fluorescent protein is loaded into PLGA particles that will sustain its release in the eye for treating back of the eye diseases. So if you get cumulative release in vitro, you can release for over 80 days and you can maintain stability of the model protein. So what we need to do now is to take this forward and look at this in an in vivo setting. We know we can pierce excised ocular tissue in this way. We need to show that we can deposit these tips in a pain-free manner in the eye, and that would sustain the delivery of a potent protein. Here you wouldn't need to have much protein in each particle, and that means that the biodegradation rather than the drug loading would control the release over time. So to summarize on dissolving and biodegradable microneedles, what we found was that only the drug inserted beneath the stratum corneum was deliverable. So there was no drug delivered from the upper needles or base plates. And that means if you want to avoid wasting your drug, you need to make sure the drug is in the part of the needle that goes beneath the stratum corneum. If you have drug in the upper part, it will end up as a sticky chewing gum-like mess on the surface of the skin that can actually pull drug out of the skin due to cohesiveness when you would try and peel the patch off. So we've seen that long-acting drug delivery is feasible over at least a month for poorly soluble crystalline drugs in our rat model in vivo. And we think from our allometric scaling calculations that weekly delivery in adults is a possibility, and so is lymphatic targeting if you get your particle dimensions right. Biodegradable microneedle systems aren't suitable for delivery of higher dose drugs, and because the drug loading ends up controlling release. But they are promising for sustained local and systemic delivery of highly potent drugs, including proteins. But obviously, if you're going to make your microneedles in 2 layers, then you've got a more complex manufacturing process to contend with. So we move on now to talk about this technology that is unique to our lab, and that is hydrogel forming microneedles. So here the microneedles themselves don't contain any drug. Instead, they're made from a chemically cross-linked hydrogel forming material that will rapidly swell upon insertion into skin to form a continuous unblockable conduit between a drug-containing layer that sits on top of the microneedle array and the rich dermal microcirculation. These microneedles can have their cross-linked density and chemical nature controlled, and also the nature of the drug-containing layer can also be modulated to allow us to deliver much higher doses of drug than would be deliverable from a dissolving microneedle system. So we're no longer limited by what we can load into the needles or even in the base plate. The microneedles are only acting as a rate controlling membrane. Another important point is that these microneedles are removed morphologically intact from the skin. So we're not depositing bulk polymer in the skin the way we would with dissolving microneedles. So we have potential for high dose drug delivery here, and we have the potential to modulate that drug delivery. So we looked at this for numerous different molecules. So here we have some examples of small molecules: tetracycline, levofloxacin, and donepezil. And what we found was that the plasma levels were still increasing after 24 hours when we removed the patch. And the same was even true for the large protein bovine serum albumin. So we started to think, what if we left the patch in place? We obviously have enough drug in our reservoir to control drug delivery over several days. So we looked at metronidazole, kept the patch on for 2 days, and we saw constant plasma levels for that entire time. So that means we have the potential to sustain drug delivery while the patch is in place, but we could also decide to terminate drug delivery in the event of side effects by removing the patch. So we then looked at this with pramipexole. So this is a Parkinson's drug. So we either applied the patch for 1 day and then removed it, or we applied the patch for 5 days and we looked at the plasma levels. So with oral delivery, we saw the typical pattern. The plasma level went up and it largely came back down again. We couldn't measure it after 3 days. With the microneedles applied for 24 hours, after 2 days, we were unable to measure the plasma concentration. But when we kept the patch in place, we saw sustained drug delivery, although the levels had started to fall. And the reason that the levels fell here was because pramipexole is a pretty potent drug, and it's known to cause side effects in animals if you dose them with too high a dose. So we didn't want to put a very high amount of drug into the patch. So we think we can sustain drug delivery for a long period of time if we leave the patch in place. That's 5 days delivery. The microneedles were removed intact and the skin looked completely healthy after the 5 days. So we then worked with some collaborators in a company called TSRL to look at sustained delivery in a mini pig, which is a very often used dermatological model. So we applied microneedles with a drug-containing layer loaded with zanamivir, the influenza drug, and we measured plasma levels over a 5-day period, and we were able to measure consistent plasma levels for that entire 5-day period. The microneedles were removed intact after that time, and you can see the skin remains healthy after the microneedles were removed. So this is very encouraging in terms of high-dose drug delivery and being able to maintain constant plasma levels while the patch is in place, but also removing the needles intact and leaving the skin unharmed. So we then wanted to see, could we deliver a poorly soluble drug in this way? So obviously a poorly soluble drug is not going to want to diffuse through a hydrogel that's 95% plus water. So we needed to make our drug-containing layer not from a water-based polymer film, not from a lyophilized wafer, not from a compressed tablet, but instead from a waxy tablet made from a combination of high and low molecular weight polyethylene glycols. So polyethylene glycol, as you may know, has both hydrophilic and hydrophobic properties, and it's commonly used as a solubilizing agent in pharmaceutical formulation. And the idea here was that when the microneedles would be inserted, they would swell, and interstitial fluid would move up to the drug-containing layer. It would dissolve the PEG, and then both the PEG and the drug would diffuse down their concentration gradient into the viable skin layers. The benefit here would be that the PEG would keep the drug in solution by acting as an in situ co-solvent, and whenever the PEG would then disperse upon dilution in the interstitial fluid of the skin, the drug would drop out of solution and form a depot for sustained release. And atorvastatin has very poor water solubility, so that's actually what happened. So what we did was we compared oral delivery, and this is a drug normally given orally at night to control cholesterol biosynthesis, and the plasma level went up and it came back down. But when we applied the microneedles and we followed the plasma levels, we had a small peak at the beginning, but we were still able to measure therapeutic plasma levels in the rat after 14 days, and the patch was only applied for 24 hours. So even this water-based drug delivery system can be modulated to deliver poorly soluble drugs, albeit as a depot rather than continuous delivery while the patch is being applied. So we wanted to try an alternative approach, so we went back to our old friend cabotegravir, and here we used cyclodextrin to enhance drug delivery. So cyclodextrins are C-shaped molecules that have hydrophobic inner cavities and hydrophilic outer surfaces. So here the idea is that a poorly soluble drug moves into the cavity and it is solubilized. So here we made a drug-containing layer based on cabotegravir cyclodextrin complexes, and the interstitial fluid came up through the microneedles, dissolved the complex, which diffused down its concentration gradient, and then upon dilution in the skin, the cyclodextrin dispersed, the drug dropped out of solution, and we were able to follow therapeutic plasma levels in the rat for 28 days. Now, what we found in this case was that for both atorvastatin delivered with the PEG reservoir and also cabotegravir delivered with the cyclodextrin-based reservoir, the efficiency of delivery wasn't massively high. So we were delivering about 10% of the active drug, whereas normally with hydrogel forming microneedles for water-soluble drug, you've been delivering 50% or 80% of the drug across the skin. We actually think 10% isn't that bad for a poorly soluble drug, because if you think about a conventional transdermal patch, it only delivers about 10 or 20% of its active drug across the skin passively, and they're still commercially viable. So we do think that there are possibilities here, even for the poorly soluble drugs. We wanted to see, could hydrogel forming microneedles deliver a peptide? So we looked at insulin in a diabetic rat model, and we looked at blood glucose in untreated animals and also in the treated animals. And here we saw sustained delivery of insulin, leading to a prolonged reduction in blood glucose levels. Now, I don't think that microneedles would ever be suitable for insulin delivery, because we can't tailor the dose the same way we can with an insulin pen or an insulin pump. But it does show that a peptide can be delivered in a sustained fashion using these sustained release hydrogel forming microneedles. Insulin has a molecular weight of about 6 kilodaltons, so quite a big molecule. We then looked at long-acting antibody delivery. So we looked at bevacizumab, which has a molecular weight of 250 kilodaltons, and we compared that with two different doses in the hydrogel forming lyophilized wafer combination with dissolving microneedle. And what we found was that we were getting measurable amounts of the drug in the plasma for over 160 hours after a single 24-hour microneedle application. So even large biomolecules can be delivered in this way. We then looked at some nanobodies that are extracted from camelids, and we had one that had a half-life extender, so it was coupled to a protein, the other one that didn't have a half-life extender, and we saw sustained plasma levels of those measured in the rat over a period of time. So even large biomolecules can be delivered through this hydrogel matrix. What we did see with bevacizumab was that absorption by lymphatic vessels was evident, and this stands to sense because the blood vessels in the skin are not going to be able to absorb a compound with a molecular weight of 250 kilodaltons, as in the case of bevacizumab. So the drug will enter the systemic circulation indirectly by first going to the lymph and then dumping out into the vena cava. But this is a possibility now if we wanted to target antibodies to the lymphatic system for immunotherapy, for example, while reducing overall whole body exposure. Microneedles might be the way to go. I talked a lot about applying large patches, so we need to be able to show that a person can apply a large microneedle patch and not use an applicator, because if each of these little 1 square centimeter microneedle patches needs 10 newtons to insert into skin, then we're going to need to apply 160 newtons here. That's going to be far too much, going to cause pain and bruising, and the applicator is going to be like a jackhammer. So instead, we need some sort of method to give a person feedback. So what we did was we used pressure responsive films that would change color if and only if 20 newtons per square centimeter would be inserted, when we knew only 10 newtons per square centimeter would need to be applied for microneedle array insertion. So we got 10 volunteers to apply the microneedles, and we compared the insertion depth for this multi-patch, that's 16 individual small patches put together, and then one small patch of 1 square centimeter. And we looked at the depth of insertion and the width of the pore created using optical coherence tomography. We found there was no difference. And the volunteers all liked the pressure responsive film, because if one little corner didn't turn red, they knew then to apply more pressure at that part of the patch. So we think that people can apply large patches, and this opens up high dose drug delivery to patients and could potentially open up the transdermal delivery market. With hydrogel forming microneedles, we think this is something that's going to be applied regularly to people's skin, so we need to show that that's safe. So we got 10 volunteers, we got them to apply microneedles to their skin every day for a week. And what we could see when we removed the patches was there was some erythema due to these tiny little insults to the skin, and this is well known in the microneedle field. And the transepidermal water loss, a measure of skin barrier function, went up immediately after patch removal in each case, but then it came back down again. And that shows that skin barrier function is restored when we came back the next day. So we then measured biomarkers at the beginning of the study and the end of the study, and we looked at IgE for allergy, C-reactive protein for infection, immunoglobulin G for an immune response, and tumor necrosis factor alpha for inflammation. And we saw no significant difference across our volunteers after the microneedles have been applied every day for a week. So we think that repeated application of hydrogel forming microneedles is going to be safe in people, and we've actually borne that out in a more detailed study that we did in the gold standard dermatological model mini pigs, and we published that work recently. So we need to be able to make a patch to be able to produce a product. So we think it would be a setup something like this, where the patch that would be worn for a long period of time would need a bubble casing to protect it, much like the Freestyle Libre, and that we would have the microneedle array and a meshed aid microneedle insertion, but protect the reservoir from being damaged, and also would allow diffusion of the drug. So we think something like this would work well to guarantee microneedle application and to keep the patch in place for sustained drug delivery. So in terms of next steps of development, I think microneedle systems have a lot of potential for long-acting and high-dose delivery of a wide range of molecules, both hydrophilic and hydrophobic, large and small molecules. And I think what I've tried to show today, at least, is some flexibility in formulation to suit individual molecules. What we're seeing in our group now is a lot of projects coming in from big pharma companies as well as smaller companies. So industry are interested in drug delivery with microneedles, not just vaccine delivery. We're working on a program funded by the UK's Engineering and Physical Sciences Research Programme at the moment. And what that is doing, as I mentioned with the mini pigs, is to de-risk the platform. So show the platforms themselves are safe, in accordance with regulatory device, and this then develops a bridging program so that a company coming along only needs to show that delivery of their drug gives them the pharmacokinetics that are desired without some side effect that wasn't seen with other routes of administration. We have seen with a number of companies, but with one in Europe in particular, a lot of progress made with scaled up manufacturing, GMP manufacture, to enable clinical trials and future mass production. And for our own work, we have 2 clinical studies that are now scheduled in collaboration with 2 pharmaceutical companies. And this is exactly what we need to see now to get patient benefit. I think historically a lot of companies maybe thought of microneedles as a developing world vaccine technology that nobody would pay them for, and so they wouldn't make money. But now I think an increasing number of companies are seeing that there are numerous drugs that can be delivered with microneedles, like maybe the GLP-1 drugs, where there are clinical advantages. The same would be true for the therapeutic antibodies, and maybe delivering antibiotics across the skin would allow you to bypass the gut microbiome and prevent resistance development. Similarly, controlled delivery of antihypertensives may maintain therapeutic plasma levels to counteract the rise in cortisol production in the morning when most heart attacks and strokes happen. So pharma companies are now investing. We have manufacturing capability in the industry, and I think companies see that they can actually make money from this, and that will be to the benefit of healthcare organizations, but also importantly for patients as well. So this is just my conflict of interest statement to let you know that I work with several companies in this area. I've out-licensed technologies on very distinct technologies to them on a case-by-case basis, but none of those companies had any input in any of the studies that I presented today, and they don't have any role in the development of the presentation that I've presented either. So these are just a list of the references that I have used in putting the talk together, and I hope that this talk has been of interest to you, and I am very happy to take some questions. Thank you very much for your attention.
Thank you so much, Ryan, for an excellent presentation. And yeah, if it's okay with you, we're going to take some questions now. So the floor is open, and I see that we already have a couple of questions in the Q&A section, and I encourage others to also share their questions. We'll try to get to as many of them as possible. So I have a question here. I'm wondering if formulation requirements are different for microneedle-based LAI. What would be a desirable formulation for this type of injectable, and any excipient that if we have would add value or make it easier to formulate?
Yeah, it's a really good question. So I think if you're going to do a long-acting injection based on microneedles, then you need to try and minimize your patch size. You don't want a patch that's enormous because it's never going to be commercially viable. So that means you need to load quite a high amount of drug into the needle tips. Maybe the needle tips need to be 80% weight per weight drug. So that means any excipients that you use have to be really good film formers so that the needles will actually penetrate the skin without essentially turning into a pile of powder and not being able to be inserted. So you need a good film forming polymer with a low glass transition temperature, but it needs to be something that is either biodegradable, so like hyaluronic acid or dextran could be a good example there, or if it's a carbon-carbon backbone synthetic polymer, it has to have a molecular weight less than about 60 kilodaltons to allow it to be cleared by the kidneys so that you don't end up with accumulation of polymer in the person's skin or in some other part of the body. Because remember, by definition, dissolving microneedles will deposit polymer as well as drug in the person's body.
Brilliant. Thank you very much, Ryan. Next question is, what about applications of microneedles for melanoma? Is that a good choice?
Well, I mean, here's the thing that I would worry about. There are lots of non-melanoma skin cancers, like basal cell carcinoma, for example, where you might want to take a conservative approach to management. So maybe administer photodynamic therapy or something like a micromode so that you reduce scarring and you still clear the lesion, because that does not have a metastatic potential. Whereas with melanoma, the challenge is that if you don't treat that appropriately, then the cells can spread and you can actually kill the person in that way. So I think really you should be excising the melanoma as soon as it's detected with a good surgical margin, regardless of scarring. Now, the microneedles may be useful after that to administer some treatment that would mop up any cells that you didn't get. But realistically, if you think that you haven't got all the cells, you're probably taking a belt and braces approach and giving systemic chemotherapy to the person, and you're not taking the risk that you've allowed that cancer to spread beyond the skin.
Yeah, makes total sense. Another question we have here. Long-acting delivery is often framed as a compliance solution, as you mentioned, especially in the beginning. In your view, is improved adherence the main driver, or are there deeper pharmacological, or public health advantages that make long-acting systems transformative, really?
Yeah, well, it can be. So we know that if we're delivering a drug orally, that we tend to often overdose the person, because we want to have a once daily formulation. And we also know that a lot of drugs are subject to first pass hepatic metabolism when given orally. So we tend to give more drug than we need. And that means we end up having either a peak of the level of the drug itself, and it'll then fall off, so you only have to give the drug once a day, and we'll peak into the toxic level. Or if in the case of high first pass metabolism, we have to give more drug than we need to get enough drug into the systemic circulation, and so we end up with forming potentially toxic metabolites that can cause side effects. So sustained delivery, whether it's an implantable system or a long-acting injection or a microneedle system or an intravaginal ring, can avoid those peaks in plasma level. And in all of those cases, your drug is going straight into the systemic circulation. It's not going near first pass metabolism. So you can minimize that production of metabolites during that first pass through the liver. And then you may also have certain conditions where you wouldn't want to have high peaks and low troughs. So maybe for management of some infectious diseases, you would rather have a constant plasma level rather than have very high levels and then very low levels. Now, that's not always true with antibiotics, of course, but for other antimicrobials, you may want to have a sustained level and don't have troughs where the infection could essentially recover. And then for something like an antihypertensive, if the person's only taking that once a day, the drug level is at its lowest first thing in the morning, just when blood pressure is rising due to the cortisol rise. Whereas if you had sustained delivery of the antihypertensive, you would then not have that problem, and you might reduce the number of patients who are having heart attacks and strokes. So there could be some benefits there as well. So I do see that it's more than about convenience. There are benefits in terms of patient care as well.
Yep. Thank you very much for that, Ryan. Next question is, if we want to restrict an LNP mRNA payload strictly to the local delivery site and avoid systemic trafficking, what are the most critical design parameters for the microneedle system/cargo? And also, have you ever evaluated any RNA, such as circular RNA payloads, in your long-acting delivery models yet?
Yeah, so that's an excellent question. And I suppose in many ways, the microneedles are only really a tool to get the therapeutic particle into the viable skin layers. So in that case, you would have to use dissolving microneedles because you can't easily deliver particles through a swollen hydrogel. So then you're having a situation where it's really the particle that's controlling where the drug goes. And I imagine that if that solid lipid nanoparticle has a propensity for being taken up by cells, it'll for sure be taken up by antigen presenting cells at the very minimum, maybe macrophages, dendritic cells, Langerhans cells in the skin. and they will then migrate to the draining lymph node. It's possible your payload may get out then into the blood when the lymph dumps out into the vena cava, but I'm really not particularly sure. I think, you know, anytime we've done particulate drug delivery with microneedles, it's been the properties of the particle that have controlled where the drug has went rather than the microneedles. The microneedles were only a tool to get the particles into the skin, and then those particles are going to do whatever they're going to do. We had a collaborator who we've worked with quite closely over many years, and she was interested in DNA and RNA. So we've done at least some DNA delivery into the skin using cell penetrating peptides, and it does seem to allow transfection of cells, but we personally have never looked at solid lipid nanoparticles or RNA. But I know a number of companies are looking at that now, as well as researchers in other groups that are more focused on things like vaccine delivery than we would be.
Great, thank you, Ryan. We have quite a few questions, so I'll try to speed up through them.
All right,
brilliant. No, that it's really good to see those questions. So we have another question, which is, can you share more about the progress on manufacturing scalability? Are you able to fill the drug product via typical sterile manufacturing fill/finish lines with minimal modifications?
Yeah, so obviously we're not a manufacturer ourselves. So whenever we're making microneedles, we make them in fairly small batches, and we use compressed air and vacuum to fill the molds. But we do work with large transdermal patch manufacturer who have a GMP microneedle manufacturing license that enables clinical trial supplies. So typically a company working with us, after we've done the animal pharmacokinetic studies, they will then go to this company and have GMP supplies made for clinical studies. So they are making the product in a low bioburden environment. My opinion, and they agree with me, is that microneedles don't need to be made sterile because the skin is not sterile. You have microorganisms right down as far as the dermis in the skin that are important actually for maintenance of skin and body health. But what you would do with your microneedles is you would design them and produce them in the same way as you would, I think, a conventional transdermal patch that has to also have a low bioburden with exclusion of certain noxious microorganisms and would have endotoxin levels below a certain amount. What we've actually found, and the company have found this as well, is you can load all the microorganisms that you want into microneedles, and then when you dry them, the bacteria, fungi, and even spores don't survive the drying process because you're driving off all the free water. So you're essentially desiccating the cells and the spores, and then you're concentrating your polymer and any drug that's in there. So every clinical trial that's been done on microneedles, and this has involved thousands and thousands of patients at this stage, has had non-sterile microneedles, and there's been no reports of any infection. So I think we can be quite confident that we're not going to need sterile manufacture here, and I think that the regulators will be convinced by the data that the companies that we and others are working with will show them that really shows that safety of microneedles, and that low bioburden manufacture is probably going to be the way to go.
Brilliant. One last thing to think about. Our next question is, many molecules are sensitive to temperature and lead to degradation due to some physical changes. How do microneedles help in this case?
Yeah, so this is obviously, we know with vaccine delivery that the cold chain is one of the grand challenges for human health, and overcoming that is a really big challenge. So the benefit of microneedles is they're a dry state formulation. So you're going to have better cold, you're going to have better storage than liquid vaccine, for example. You're also not going to have to reconstitute it with anything. And if you look at some of the work done by Micron Biomedical with their measles and rubella live virus vaccine, based on dissolving microneedles, they've had really good thermal stability. And it's because most chemical reactions don't occur in the dry state. Ions, atoms, molecules aren't as free to move, and so you don't get the same reactivity as you would in a liquid environment. So I think that microneedles have a big potential to take the cold chain out of the equation, and particularly for vaccines in developing countries, and that could be a massive benefit to millions and millions of children in those countries in particular.
Brilliant. Our next and I believe last question is, thank you for the excellent and insightful presentation. What is the practical maximum microneedle patch size for clinical and commercial use? And how do age, body mass index, reflecting differences in subcutaneous fat and skin mechanics, and body site as well, influence the feasibility and effectiveness of large patch applications?
Yeah, it's a brilliant question and really important from a translational point of view. So the biggest microneedle patch we've ever made is 30 square centimeters. That's not to say that a bigger patch can't be made. What you surely need is a feedback mechanism that shows that the majority of the needles at every part of the patch have been inserted, because otherwise you're not going to have a project or a product that's going to be reliable from one patient to the next, or even in the same person from one application to the next. That's important. And you're right, body site is really, really important, because the amount of muscle and subcutaneous fat will vary from one person to the other. So what I think is that microneedles will be applied to similar sites to where we apply conventional transdermal patches. So they'll be applied to the shoulder, underneath the collarbone and hip region. And you would hope that there would be not so much variability there. We've just done a study actually with human volunteers, and it showed pretty consistent insertion at the 3 different sites, at least for small microneedle patches across human volunteers. But obviously this would need to be looked at for bigger patches and in a greater age range than we were able to look at in a small study here. But I think that what's important as well is that we have shown that repeated application of microneedles doesn't seem to cause any issues with the skin. And this is important. Now, I still think you would recommend to your patient to rotate the site of application, more so because of the adhesive that would be used to keep the microneedles in place, I think, rather than the microneedles themselves. So I'm quite encouraged by what we've managed to achieve so far, but I think we still have a little way to go before we will have a microneedle product on the market that is going to benefit patients. But ourselves and others and industrial colleagues are all working hard to try and push that forward to try and achieve benefit for patients.
Brilliant. And on that very positive note, I think that we're right on time here. So I would like to extend a very, very big thank you again to Professor Ryan Donnelly for your excellent talk today, and for all of you for joining us. Just to say, a recording of today's webinar will be free to access on the AGS Talks website in the coming days, and we will also email you all a link. You're also welcome to share the link with any friends or colleagues who are not able to attend today. As I mentioned, the event is sponsored by the Biomedical and Life Sciences Collection, where there are thousands of talks across different scientific fields from leading experts, including pharmaceutical developments and others. By all means, have a look at the resource tab on your platform here for additional information. And my email is also in the chat if you would like to contact me or with any question about the collection, or if you would like some access to the collection, we can arrange that as well. Thank you again so much, Professor Donnelly, for presenting, and thank you all of us for joining us today, and I hope you have a wonderful evening or day, depending on where you are, and we'll see you on in future events.
Thanks, Eyal. Thanks, everybody.
Thank you very much. Take care, everyone.
Thank you. Bye.
Bye-bye.